Controlled human infection model is a type of clinical trial where you get a certain pathogen, so a bacteria or virus, you introduce it to a healthy individual, a healthy volunteer, after giving them a vaccine. Controlled human infection model studies are important because they enable you to answer a question about effectiveness of a vaccine using a smaller number of participants in a relatively shorter period of time. When we give the vaccine to a few hundred people in the trial, we also give another group a placebo. A placebo is a substance that has no treatment value. Both groups, so those who received the vaccine and those who received the placebo, go on to be challenged with an infectious agent. We then compare how many people carry or develop a disease in these two groups. So this gives us an idea of how effective the vaccine is. This platform can be applied across various diseases or various vaccines. So for malaria or typhoid.
Pneumococcus is a bacteria which commonly infects all age groups. What we are doing is basically to see if the vaccine can help to minimize or reduce the bacteria being found in the nose.
PCV13 is, the full name is pneumococcal conjugate vaccine. So this is a vaccine that is given to children under 5 years here in Malawi. This bacteria is quite notorious for causing pneumonia, so lung infection. It also causes ear infection and can also cause meningitis, so in severe cases. For you to actually get an infection, you must have this bacteria in your nose. After introducing this vaccine, PCV13 in Malawi in 2011, as scientists, we've been closely studying how effective it is, how good it is in our population. So the pneumococcal conjugate vaccine is good at protecting you, but it's not been as good as protecting you from transmitting the bacteria to the next person.
We're interested in the bacteria called Streptococcus pneumoniae, also called pneumococcus. And for our type of human infection study, we very carefully select healthy volunteers at very low risk of developing any disease or any problems, and we expose them to the bacteria. And what we do is just gently put the bacteria in their nose and allow that bacteria to grow in their nose. The participants don't become unwell during that process, but just in case, we very carefully monitor them and provide clinical cover, such as me, to ensure that no problems happen. So this type of work is helpful for us to understand how the disease works, how the body reacts to the bacteria, and also to try to find better vaccines. If the bacteria doesn't grow in the nose, you know that the vaccine's worked. If it does grow in the nose, then the vaccine hasn't worked, okay, in very broad and simple terms.
So from the time they go into an agreement with the study team, we request if they are comfortable for us to collect samples. We are collecting a number of samples. All the samples are safe. For example, we collect what we call a nasosorption sample. We measure the immunity of the participant at the time they are joining the study. The next step, we collect what we call a nasal wash. Why we are collecting the nasal wash is to see if the participant is already carrying the bacteria we are interested in. The next sample we collect is what we call a nasal scrape. In that as well, we are also doing different tests to see how the immunity of the participant is looking like. From the nasal scrape, the next sample we do is a throat swab. This is basically a COVID test. It's not really primarily part of the study, but it's a safety precaution because we understand that we are living in the COVID era. We want to make sure our participants are safe and also the study team is safe. We collect what we call a salivary sample. we are also testing the immunity. The next sample we are collecting is a blood sample, checking how the immunity of the participant is like at the time they are joining the study. After 1 month, we invite them back to the clinic for what we call a screening test. On this day, what we are doing is now introducing the actual bacteria. So this is when the experiment now begins. We give the participants amoxicillin. This is an antibiotic just to cover them in case they get sick from the bacteria that we introduce in their noses. We also have two other safety precautionary measures. Number one, we have an agreement with Malawi Hospital to make sure that should the participant fall sick, which we haven't seen so far, but then things could happen. They have to get the best medical care as per the Malawian standard. We also offer them what we call a residential stay at Grace Bandawe Conference Center. This is to make sure that we know where they are, but the study team is also available on site with the participant. Should the participant get sick at any point from the time we introduce the bacteria, the study team is there to intervene. How? If the participant is carrying, then we offer the participant to take the antibiotic. If the participant is not sick, but they are carrying, we allow them up to the end of the study. If they are carrying the bacteria we introduced at the end of the study, then as a study team we also come in to give the antibiotic so that we can clear out their nose. Remember, we started with a clean participant who did not have the bacteria in their nose. We also make sure that they are safe during the entire study period. And if they are carrying, we still allow them to also go with a clean nose by offering them the antibiotic.
The problem with the pneumococcus, this type of bacteria, is that most people who are infected with it are actually from sub-Saharan Africa. And the vaccines which are developed were developed in higher income countries, and they seem to work quite well there. But in Malawi, whilst they protect the person who's been given the vaccine from developing their disease, the herd immunity effect is less. What I mean by that is that the vaccine should stop the bacteria from growing in the nose. Okay. If the bacteria grows in the nose, then other people who haven't been vaccinated are at risk of contracting that bacteria. And if they are vulnerable, so if they are elderly or if they have diseases such as HIV infection, for example, they are then at high risk of developing invasive or disease through this bacteria.
We've been conducting this research at Malawi Liverpool Wellcome Trust since 2019. Because this was the first of its kind, we had to perform the study in a smaller number of participants. So we did get all regulatory approvals from our institutional review boards, so COMREC, NHSRC. They looked at our protocols, they looked at our standard operating procedures, the safety measures that we had in place, and they saw that we have the capacity, we are ready to conduct such type of a clinical trial. We have recruited over 120 participants in our study. We have given the bacteria to over 70 participants. And so far we haven't had anyone admitted to hospital as a result of this study.
And the problem with vaccines is that they're very, very expensive to develop, and this this method, this way of choosing vaccines helps us to have confidence in a vaccine before we then move to the large-scale clinical trials.
My fears were overcome due to the second visit which I came to the clinic, to hear more about the bacteria and the vaccine. Like, we had a number of questions which I asked to the nurses and the doctors which were there for the information visit. So all the questions which I raised on that time were answered clearly, and I was convinced that it's safe to do the study.
We are looking for adults from the age of 18 to 40 years of age who are healthy. We are inviting them to our research clinic. First of all, discuss with them or give them information about the study. From there, we give them some time, say for example 1 week, to make their decision. Once they have understood and they are happy to come back, we invite them back to the clinic.